Good day, dear students. Today, we will be discussing about the steps in scientific method and the three types of variables. To start, let's define the word scientific investigation. Scientific investigation is a process used to find answers to questions about the things around us. It is often following a general pattern, and that general pattern is what we call the scientific method. In the past few lessons, we have mentioned that scientific method is the step-by-step process of solving problems. In other words, it is a series of steps that we can use to solve or answer a problem. It is also a pattern or body of techniques used to investigate a phenomenon, acquire new knowledge, or correct and integrate previous knowledge. Since scientific method is a process, we cannot proceed to the second step without doing the first step.
All the steps of scientific method are important.
Therefore, we should not disregard any step that we will mention here. The first step of scientific method is observe and identify the problem. In order for us to know the problem that we have around us, we need to observe or to use our five senses. These problems should be written in question form. Formulating the right question is important, but not every question is a good question. A good question is one that can be answered through investigation, not by reading a book or researching the internet. Look at this picture. What problem can you see in this situation? Yes, the problem is
There are so many garbage on the road.
we were able to identify the problem because we look at the picture. In the example, the question form of the problem given will be, why are there so many garbage on the road? Let's try to identify the good questions on the following numbers. In your notebook, write 1 to 5 and put a check mark on questions that are considered good questions to be studied and answered through scientific method. If not, put an X mark. You have 2 minutes to answer these exercises and your timer starts now.
Let's check your answer. For number 1, Which will germinate faster, mango seed or mongo seed? The answer will be a check mark, because we can make an experiment and perform a scientific investigation for this question. Number 2, where do foxes live? The answer is an X, because this question can be answered by reading a book or researching the internet. Number 3, what type of soil produces healthy plants? Number 3 is a check. We can perform scientific investigation for this question. Number 4, what will happen if you do not have dinner? It is a check mark. And number 5, what happens after we die? Number 5 is an X, because after we die, we will not be here anymore to finish the scientific investigation. What score did you get from these exercises? If you got a score of 3 and below, ask your teacher to explain this further to you. If you got a score of 4 to 5, very good. You did a great job. Let's now proceed to the next step. The second step of scientific method is to conduct or perform a background research. This step can be done by asking the experts on topics related to the problem that you want to answer, reading books, and searching the internet. Just make sure that the information that you get is reliable. Step number 3 is forming a hypothesis. Hypothesis is an educated guess, meaning it is based on your research. From the research you have done, what do you think is the temporary answer for the problem that you wanted to solve? You can formulate a hypothesis as an if-then statement. For example, the problem that you wanted to investigate is the question, which will germinate faster, mongo seed or mango seed? After your research on factors that affects the rate of germination, you will now give your tentative answer or your prediction to what will happen in your investigation. Your hypothesis might be, if I plant mongo seed and mango seed under the same condition, then mongo seed will germinate faster. The next step of scientific method is to test the hypothesis or conducting an experiment. In this step, you will try to find out if your hypothesis is valid or not. You design and perform an experiment to test the hypothesis by preparing the materials and by giving the procedure that you will follow in doing your experiment. In conducting an experiment, you need to identify the 3 important factors, or what we call variables. The 3 factors or 3 variables are: number 1, independent variable. This refers to the quantity which is identified as the possible cause of change. It can be manipulated. This is the factor that you, as the experimenter, manipulated or changed so that you will get the answer to your problem. This is also called the manipulated variable. Number 2, dependent variable. It is the factor that changes in response to the independent variable. It is also presumed as effect. It is the factor that changes because of the change that you did to the independent variable. Number 3, the control variable. It is a part of the experiment that is not being tested and used for comparison. In other words, it is the constant. For example, the problem that you wanted to investigate is, which will germinate faster, mongo seed or mango seed? The hypothesis that we have is, if I plant mongo seed and mango seed under the same condition, then mongo seed will germinate faster. Now, let's try to think of the materials that we need to prepare for this experiment. Of course, we need to have the seeds. We are going to have 2 different seeds, which are the mongo seeds and mango seeds.
Next, from the problem that we have, what do you want to know? We want to know which among the seed will germinate faster. In other words, you wanted to know the rate of germination of the 2 seeds. And lastly, the other materials that you will need for this experiment are the soil, the pot, For this experiment, we will be using the plastic pot so that we can see the development that is happening on the seeds. The amount of water and the exposure to air. Based on the materials and things that you will examine on your experiment, what do you think is the independent variable? Yes, the independent variables are the types of seeds because we have two different seeds, the mongo and the mango. While for the dependent variables, we have the rate of germination. And the control variable will be the types of soil, the plastic cups, the amount of water, and the exposure to air. Next step, or the step number 5 of the scientific method, is record and analyze data. While conducting your experiment, you need to record the data and then analyze it to get the answer for your experiment or for your problems. Data are the results of your experiment. This data can be recorded through graphs, tables, and diagrams. An example for this is the following. Assuming that we have done with the experiment testing the germination rate of mongo and mango seeds, we will have this diagram. For day 1, both the mongo seeds and mango seeds showed no change. For day 2, no change at all between the mongo and mango seeds. For day 3, mongo seeds started to break, while mango seed showed no change at all. For day 4, mongo seeds developed small leaf. Mango seeds, there is no change. For mango seeds, during the day 5, roots, stem, and leaves are now observed. For mango seeds, there was no change. For day 6, in mango seeds, roots, stem, and leaves are growing. For the mango seeds, there was no change. And lastly, for day 7, for mango seeds, new leaves are observed, while in mango seeds, that seed started to break. For the last step of scientific method, we have a draw or forming a conclusion. If hypothesis is an educated guess or a tentative answer to the problem, conclusion is the final answer to the problem based on the outcome of your experiment. You can make a conclusion based on the data you have gathered. So, based on the data of the experiment done about the problem, which will germinate faster, mongo or mango seeds, what conclusion can we make? So we have the problem, which will germinate faster, mango seed or mango seed. The hypothesis, if I plant mango seed and mango seed under the same condition, then mango seed will germinate faster. And the data that shows the development of mango and mango seeds from day 1 to day 7. Based on the data gathered, the conclusion will be, under the same condition, mango seed will germinate faster than the mango seeds because the mango seed started to develop into a small plant during the day 3, while the mango seed change was observed during the day 7. So we were done with the steps of scientific method. Number 1, observe and identify the problem. Number 2, perform background research. Number 3, form a hypothesis. Number 4, test the hypothesis. Number 5, record and analyze data. And number 6, draw a conclusion. Now that we were done with the steps of scientific method, you can now answer the remaining parts of your module. From Activity 1, Activity 3. Activity 3.1 and other pages from page 8 to page 10. Again, if you have any questions regarding the lesson or your module, contact your teacher right away. So dear students, you may now start answering your module. God bless.